Hey guys, welcome to this presentation on making Punnett squares and Punnett square analysis. Before we begin, I just want to reinforce where alleles are, how genes relate to DNA and chromosomes. So here we have a picture of a chromosome, and on that chromosome are different genes. This is a gene, this is a gene, this is a gene, and all those genes code for a specific trait. They each code for a different specific trait. And then if we look here at these chromosomes, again, these are all different genes. Okay, and this shows this zoomed in onto the chromosome, and you can see the DNA double helix here. Okay, and then if we zoom in even more, we see the nucleotides and the nucleotide sequence. And you have to remember back to protein synthesis that the nucleotide sequence codes for a specific trait. Okay, the nucleotide sequence on a gene will give a specific protein. Remember that proteins are what pretty much do everything in your body. Okay, so between two like chromosomes, two homologous chromosomes, we have the same gene on each of those chromosomes. However, the alleles for them could be different. Okay, so this chromosome has the allele for blue eyes, while this chromosome over here has the allele for brown eyes. So genes have to be in the same spot on homologous chromosomes, but the alleles can be different. The allele is the type of characteristic that the gene is coding for. And here again, we have a picture of a chromosome, and when we unwind that chromosome, we eventually get down to our DNA double helix. And that DNA double helix has the nucleotides. And remember from transcription and translation that those are the directions for making a protein. Okay? Each of these three triplets makes a codon on the mRNA, and that will code for specific amino acids, link all those amino acids together, and we get a protein. When making Punnett squares, I always want you to remember that whatever genotype you receive, that is how you're going to determine the phenotype. Now I want you to take a few minutes and watch this animation on how to do Punnett squares, and it gives you a perfect example of how Mendel did his Punnett squares.
Gregor Mendel conducted experiments to answer one of the fundamental questions of heredity. What are the basic patterns in the transmission of traits from parents to offspring? In this recreation of Mendel's historic experiments, you will have the opportunity to predict the outcome of two types of genetic crosses using a Punnett square. In this first example, the plants being mated differ in just one trait. One plant is homozygous for the dominant purple flower allele of a gene. The other is homozygous for a recessive white flower allele. To show the mating, we arrange the parent plants along two sides of the square. Note that each parent produces a single type of gamete. The genotype of each gamete produced by one parent, shown on the top, is brought down into each of the squares below it. Likewise, the genotype of each gamete produced by the other parent, shown on the left, is moved into each square to the right. The Punnett square helps us predict the genotypes of the offspring. Using the information provided, predict the phenotypes of the offspring. Drag the correct phenotypes to each square in the grid. Notice that all of these offspring have the same flower phenotype as the purple flowered plants in the parental generation. In the offspring, called the F1 generation, the white phenotype disappears. If we mate these heterozygous plants to each other, or allow them to self-pollinate, what kinds of offspring would you expect? From the choices given, select the correct gametes to place on the two sides of the Punnett square. Using the information provided, predict the genotypes of the offspring. Drag the correct genotypes to each square in the grid. Using the information provided, predict the phenotypes of the offspring. Drag the correct phenotypes to each square in the grid. This first series of crosses recaps the results of a mating in which the parent plants differ in a single trait. The parent plants are homozygous for the dominant and recessive alleles. Their F1 offspring are heterozygous and show the dominant purple flower phenotype. The next generation, called the F2 generation, reveals a typical 3-to-1 ratio of phenotypes. The recessive white-flowered phenotype reappears in this generation after having disappeared in the F1 generation.
A Punnett square is a model that predicts the likely outcomes of a genetic cross. It is named for its inventor, Reginald Punnett. Here's a picture. And it shows all the genotypes that could result from a given cross. And that's how we're then able to determine the phenotypes, possible phenotypes. So we start by making 4 boxes. We put the possible alleles from one parent along the top, and we write the possible alleles from the other parent along the side. Each box inside will hold 2 letters, one from each parent, and each box will represent the possible genotype of the offspring. And whatever genotypes we get in those boxes can be analyzed, and we need to identify each of them as homozygous or heterozygous and dominant or recessive. So if it's homozygous, the letters are going to be the same. In other words, they're always the same letter, but it's whether they're big or small that matters. So here we have 2 big letters, and here we have 2 small letters. But because these are both big, they're homozygous. Because these are both small, they're homozygous. Notice here that we have 1 big and 1 small. So that's why they're heterozygous. We have a big and a small. So then we need to identify it as dominant or recessive. Dominant is represented by a big R. Okay? Big R's are dominant. Recessives are represented by little r's. So the reason that this one down here is dominant, even though it's got a big R and a little r, the big R is present, and therefore that's what makes it dominant. So finally, just a quick overview of the steps of making a Punnett square. First thing we need to do is identify the parents. Then we need to do the cross. Then we need to identify the possible genotypes, and that will be what's inside the squares. Identify the percentage of each type of genotype. Identify each genotype as homozygous or heterozygous, and then as dominant or recessive. And then finally, identify the phenotypes based on the genotypes that are produced.